Chandrajeet Kumar, the first presenter, on vermifiltration of arsenic contaminated water using vermifiltration technology, a novel biofilter model. You have 7 minutes. 7 minutes.
Person, respected chief guest, and the chairman and the organizing committee of this university. A warm good morning to all of you. So I am going to start my. Sorry, I forgot to introduce. This is Chandrajeet Kumar from N College, Patna. Recently, I have submitted my PhD thesis last year, and some part I am going to share with you all. My topic is that the vermifiltration of arsenic contaminated water using vermifiltration technology, novel biofilter model. Next slide. First of all, I would like to introduce my research team. I am along with Dr. Ashu Ghosh. Right now, he is the chairman of Bihar State Pollution Control Board. And one girl is there in the red block, Susmita. She was with us last year. Unfortunately, she died and we lost her. She was directly associated with this research. That is why I am putting her name here. Next slide. So my main research objective is that how I made a vermifilter unit to purify the sewage water and the chemically contaminated water, and for that I purify the arsenic contaminated water. Next slide. So first of all, I would like to introduce about what is vermifiltration technology. This is the simply innovative wastewater treatment and purification technology that detoxify and disinfect by earthworms. As we know, the Sir Charles Darwin first of all told about the earthworms that are the unrivaled soldiers for the mankind and the friends of soils. So earthworm converts wastewater into nutritive water that is full of nitrogen, potassium and phosphorus. And the purified water or treated water can be useful for the agriculture, irrigation and the non-potable purposes like your cleaning your homes, gardening and the washing your cars and the toilet flushing etc. The main researchers that works on the vermifiltration technology is that are the following. First of all, the vermifiltration idea came from the Professor Maria Soto Chile, and after that the Professor Dr. Rajiv Kumar Sinha, photographs are available there. He is a mentor of my mentor, currently working on the vermifiltration technology and made a vermifilter unit. And in India and Baroda, Transchem Agritech is a company that is commercialization for the vermifiltration unit. Next slide. This is my vermifilter unit that is very cheap and best. The feature of that, this is the zero waste technology and self-promoted, self-regulated, self-improved and self-enhanced. Easy to operate and runs without electricity. And this is totally aerobic process having no odor, no slush, and of course there is no chemicals are used. And the main thing is that by this filter we can use liquid as well as solid waste. And the main thing is that we can treat sewage water and industrial influent from that. Next. This is the microscopic view of my filter. These are the systematic arrangements of stone chips, and the top layer is the sawdust and the sand, and the toppest layer is that the earthworms that occupies with the soil and the vermicompost. These are very simple but very useful for the student, researcher and of course for the farmers. Right now this is in Patna, 2 to 3 colleges they are applying for this vermifilter unit, and I am trying to base my level to spread this for the other schools, colleges and the homes and domestic uses also. The earthworm's body acts as a biofilter that removes sewage, BOD, COD by more than 90%, TSS, TDS by also 90%, and of course the greenhouse gases becomes reduced by 99%. And earthworm also accumulates endocrine disrupting chemicals, heavy metals, toxic chemicals, and earthworm's body secretes coelomic fluid that having anti-pathogenic properties that kills all the E. coli and removes 99% fecal E. coli forms. Next. These are the vermifiltration field that is Baroda, and this is the Ganga water before filtration and after filtration. We can see the lots of changes. Next. The main thing I filter the arsenic contaminated water from this filter. I just prepared the two concentration 10 ppm and the 20 ppm, and finally filtered it through the Vermifilter, and the earthworm's body tissue as well as the treated water was analyzed by the AAS, atomic absorption spectrometer. Next, the result which I found that when 10,000 ppb was passing through the Vermifilter unit, the treated water having the first result is that decreases to 7.7 ppb, and when we pass the 20,000 ppb, it becomes the 6 ppb. And earthworm's body, whenever we analyze by the AAS, we can see here earthworm's body also accumulate the arsenic. That proves that the arsenic enters inside the earthworm's body, and earthworms tolerate this arsenic and converts it into the useful product, and finally decreases the value of arsenic. Next, the future objective for this is that in the pathway of vermifiltration, we will have to find out how many arsenic and which form of arsenic is going to be accumulated. And suppose if earthworm is surviving in the toxic concentration of or environment of arsenic, that is certain genes are responsible, that is providing tolerance capacity for that earthworm. And the earthworm's gut contains lots of microbes, that is the known of microbes factory. We will have to identify the role of microbes present in gut of earthworms in arsenic conversion. So I would like to conclude with my slides with my slogan to avoid using clean water. Actually 80% of fresh water is we are utilizing, so we need to recycle the water. Thanks a lot. Thank you. Essenia fetida. Essenia fetida. That is easily available in India.
What is the filtering capacity of this?
What is the loading capacity of this arsenic on this material?
In my test, I have taken the 10 ppm and 20 ppm.
Loading capacity, how much arsenic it can retain into the
Yeah, this is the result portion. If you suppose the 0.5 milligram of earthworm body tissue, we will go to the result slide. Earthworm body tissue where we kept the 0.5, it is analyzed 127.9.
So how long it will accumulate?
This.
Yes.
Actually, this whenever it reproduces, and finally they never transfer the arsenic to one generation to the next generation. Finally, it whenever we long exposure while providing long exposure to the arsenic toxicity, it becomes dye. Earthworm becomes dye.
Shall we need to store the earthworms after using it?
Yeah, you can culture the earthworms in separate bucket for your future use.
The earthworm dies because of due to intake of more arsenic. Then we have
But still till 20,000 ppb, we are seeing that the earthworms are surviving.
actually solid waste management again will come to the picture. When you are using more earthworms,
We need to reculture or to subculture or to transfer the earthworms day by day, or monthly or weekly.
you have to replace every
Yeah.
week you are placing.
Again, you are creating a lot of waste containing the arsenic.
This is where I am creating the waste. No, the waste which are you getting as the earthworm cast, you can directly keep it to the field farm as a biofertilizer.
No, but again, if you are using it as a biofertilizer, you are going to add it to soil into the field. Then again, it is reaching to the same system.
I am not getting the question.
So, if you are removing the earthworm from the system and then again adding it to the field for the biofertilizer, then again you are going to add this arsenic into the agricultural field, which is going to prevent the growth of the plants in the agricultural field also. Generally, that you are recycling the same arsenic into the same system.
No. Earthworm's body, suppose it. Will you please try to explain in Hindi?
If you are purifying the water,
Arsenic
is going to be accumulated into the earthworms. If you are again using this earthworm into the agriculture, you are adding arsenic to the agriculture, which is going to create a problem for the agriculture too. Yes.
Let me refrain the question which sir is asking. I'm from Tata Projects. Good afternoon, everyone.
Hello.
The question is, what is happening to the arsenic first? Whether it is getting converted into some non-active arsenic form, or it is getting accumulated into earthworm. And if it is getting accumulated, then what is happening with that the active biomass content of the earthworm?
This is I have kept in the future objectives. This is the point of study.
Okay.
This is the first frame of your result.
Fine.
And that is why the conclusion slides are not is not available in this
So
slide.
as of now, you are having an answer to the first question, that it is getting converted,
Yeah.
or maybe it is getting absorbed
Yeah,
by the.
I need to just identify in which form it is converting and what does it happen after the conversion.
So that means the active biomass still have that arsenic which may be into active form, or solid form, or this arsenic can still be a potential threat for the environment, even though it has been reduced by a large amount with the help of earthworm.
This is the remain part of my study, and this is for the postdoc or like that.
Fine. Another question from my side. What is the capacity up to which it can handle? You have specified one Gujarat plant is of 1200 KLD.
Yeah.
What capacity up to maximum limit it can handle, and what is the area requirement for this plant?
I can't answer to this question because I am not from the commercialization.
No.
Okay, you may interact in other session also. So now next speaker.
Okay, thank you so much.